Okay, for Wednesday, how many of you think that you are able to, I'm not saying excited about, coming in at 8 as opposed to 9 for the lab exam? I'm hoping we can get about half. Okay, well actually that's, so if you can come in at 8, that would be good. Particularly if you have an 11 o'clock class. Okay, that way you're not going to be rushed. And then if you don't come in at 8, go ahead and come in, 9, maybe even 9:30 if you don't have an 11 o'clock class, because that way we can stagger people. On the lab exam, within reason, you are not timed at a particular station. Now, if you sit for 45 minutes at the microscope, at some point in time I might say, you got to move on, okay? But within reason, I let you spend the time that you need at a particular station. But that way, it creates a little bit of pressure with this large size of class. So if you can come at 8, be here at 8, ready to go. And if you're not coming at 8, come in sometime after 9 between 9 and 9:30, okay? When you come in, there will be answer sheets for you at the corner on the end of the table by the door. I would prefer if you could leave your books and backpacks and coats and things out in the hall or in the lounge, because if we have them in the aisles, it's going to be really hard for people to move around. Short answers, okay? This is one time when I don't mind you writing in incomplete sentences, couple word answers many times will suffice. That's about all the instructions that I need to give you. If you have any questions about the lab exam, tomorrow would be your last chance to ask. Okay? So you all fired up for the rest of the semester? It's not a very enthusiastic looking response. Okay, so what we're talking about now, and we'll be talking about for a lot of the remainder of the semester, is the interaction that takes place between a host and a pathogen. And remember that when we talk about relationships of organisms, that the pathogen is considered to be a parasite, and if the parasite gains the upper hand in the relationship and starts causing disease symptoms, then we develop a definable disease state. Okay? In that particular situation of a disease state, we have an infection. And by definition, you have an infection when you have microorganisms that are growing and proliferating in what are normal microbial-free tissues. Okay? So that's the criteria. Now, is all disease caused by infection? No. There are other causes of disease, like blocked coronary arteries. We didn't, although used to think that there was any microbial association with blocked coronary arteries, but one of the surprising connections we're starting to see is that diseases that we used to think were not microbial in nature might have a microbial influence. For example, in coronary artery disease, inflammation seems to be a real important factor in causing those vessels to become blocked. And one of the things that potentially can cause inflammation are microbial infections that are subclinical in nature. You don't know you have this particular infection, but the inflammation is causing the buildup of plaque. The buildup of plaque is causing the blockage of the arteries, and the lack of blood flow to the tissues is causing the symptoms of the disease. So even things that we didn't used to think were microbial in origin now might have microbial influence, at least. The first step in the process of causing an infection is the microorganism becoming attached to the tissues. And remember, we said that the place in the body where microorganisms gain entrance is called the portal of entry. And different microorganisms are adapted to different portals of entry. And sometimes it is very, very specific. Take the rhinovirus, for example, which causes common cold. The rhinovirus has a specific portal of entry to your body, and that is the lining of the nasal respiratory tract. You can rub cold virus all over the surface of your arms, for example, and it won't infect. Why? Because those cells lack the proper receptors for those virus, in this particular case, to attach. If attachment doesn't occur, infection won't occur. Microorganisms have molecules on their surface that are also surface specific. Here's a microorganism, and sticking off the surface of that microorganism is some kind of a molecule, a ligand, and if that microorganism is going to infect this particular tissue right here that this host cell is part of, maybe this is part of a mucous membrane, for example, in your nose, then ligands on the surface of the microbe have to bind to receptors so that specific attachment will take place. Those receptors are oftentimes referred to as adhesins. Okay? So there can be adhesins on several different cell surface molecules. They might be part of the glycocalyx of the bacterium. They might be part of the pili. They might be part of the flagella. If mutations occur in the genes that encode these adhesin molecules, a microorganism can lose its ability to become pathogenic because it can no longer attach. One of the strategies that your immune system can oftentimes use to prevent infection, and this is particularly true with virus, is to develop antibodies against the adhesin molecules on the surface of the pathogen. And if the antibodies bind to those adhesins, it blocks them and prevents those adhesins from then attaching to the cells, and therefore infection won't occur. Those are called blocking antibodies, and that's one of the ways antibodies can protect you against infection, is to cover up the adhesins on pathogenic microorganisms. So, penetration into the tissues and then subsequent growth is the definition of having an infection. Let's make sure that we know a little bit about tissues before we proceed on and talk about adaptations that microorganisms have to cause them to become infected. So a little animal physiology A&P review right now. What are the four basic tissue types in the human body? Dr. Rhodes is turning around in his chair in his office right now. Mackenzie? Epithelial? Epithelial, connective, muscle, and nervous tissue are the four basic tissue types of the body. Two of those tissue types play a really important role in what we're later going to talk about as the primary defense system against infection. What is one of your important primary defenses? Intact tissue, including skin. So we're going to talk a little bit about adaptations that microorganisms have to allow them to breach this primary defense barrier of epithelial and connective tissue, which help to make up that barrier of the skin, which protects you against a remarkable amount of infection. So let's take a look at epithelial tissue just for a second. What's the definition of a tissue? A group of cells working together is one definition. Something I would add to that particular definition would be that the cells are connected to one another. So it's a group of connected cells. Working together implies a similar function. How else might cells be similar other than function? What relates to function? Structure. Okay, so it can be a similar group of connected cells that are similar in structure or function, usually both. Epithelial tissue is typically broken down into two subcategories: keratinized epithelium and epithelial mucosa. Where do we find keratinized epithelium, do you know? Skin. Okay, the skin is a classic example of keratinized epithelium. What is keratin? It's a protein. And it's a very insoluble protein that becomes deposited in the skin, particularly as the skin ages and moves toward the surface of the skin, as those skin cells move toward the surface, and eventually those skin cells are sloughed off. So deposition of keratin is oftentimes associated with skin cells that are more toward the surface of the tissue and are in the process of being sloughed off eventually. It's a protective function, okay? That keratin makes the skin cell tougher and less likely for microorganisms to penetrate across that particular barrier. Where do we find epithelial mucosa? We find it in the lining of all of the tracts of the body, including the respiratory tract, the urinary tract, the GI tract. And remember that all of those tubes that most people think of as being inside your body actually represent external environment. Okay, your GI tract is a tube running through your body. But when you ingest things, you're ingesting them from the environment, and so the surfaces of those are actually external to your internal tissues. Does that make sense? Those linings of the epithelial mucosa secrete mucus. There are specialized cells in the epithelial mucosa called goblet cells. Remember those? And they secrete this slimy substance. What's the purpose of mucus? Think about your respiratory tract, for example. Why is it advantageous for those epithelial mucosal cells of your respiratory tract to secrete this slimy substance? Right?
It's a defense mechanism.
It's a defense mechanism, and how does it work? Yeah, it is.
So you can get like carcinogens or like pathogens.
The mucus surrounds the pathogens, and what does that do? How does that protect you? Prevents attachment, okay. Covers up the pathogen, it slimes it, okay, and it makes it less able to attach to surfaces. And if another defense mechanism like coughing, for example, is initiated as a result of being associated with these pathogens or sneezing, that those slimed pathogens can be expelled more easily than ones that have grabbed onto the mucosal tissue. There's another adaptation that some mucosal tissue has, and that is that some epithelial mucosa is also ciliated. Why is that the defense mechanism? What does that cilia do? Where do you have ciliated epithelium in your respiratory tract? In your trachea. Okay. Think about all the junk you inhale and all the potential pathogens that are in dust and dirt in the air that you inhale. There's a lot of them. Well, the mucosal substance that's secreted by those epithelial cells helps to prevent attachment. Also, as the cilia beats back and forth, it creates a sweeping motion which moves the pathogens that have been coated by the mucus back up the trachea so they can be expelled by coughing or sneezing or spitting or whatever. They call that the ciliary escalator. Okay, it creates kind of just an upward sweeping motion that prevents particles from getting deeper down into your lungs. You don't want those things settling into your bronchi or your alveoli, because then it's hard to get rid of them, and lung infection is a possible outcome of that. There are a number of reasons why you shouldn't smoke. And one of the reasons is that tars and nicotine have been shown to paralyze cilia and compromise that defense mechanism. And so smokers have a much higher incidence of lung infection than non-smokers because of those effects on the cilia of your epithelial mucosa. Intact epithelium is a remarkably good barrier against infection. You just think of all the things you're exposed to and how your skin is constantly being associated with microorganisms which are potentially pathogenic. But if the skin remains intact, it is a very effective barrier. Now, we're going to talk about some adaptations that microorganisms have in a minute that it can allow them to breach that barrier. But generally speaking, unless that barrier is broken by some kind of mechanical injury like a cut, it works really, really well. That's one of the reasons why burns are so serious, is that when you burn skin and breach that particular barrier, all of those microbes that you're exposed to can now easily gain entrance into your tissues. And so burn victims have to be treated very carefully, have to be treated with sterile dressings, and put in sterile environments whenever possible, because secondary infections following burns are the primary cause of death from burns. What's the definition of a connective tissue? What makes something connective? What are some examples of connective tissue? Cartilage? Blood is the connective tissue. Blood is oftentimes referred to as an atypical connective tissue. Cartilage is considered a more typical connective tissue. What makes a tissue a typical connective tissue? You've got living cells, but typically they're embedded in some kind of a non-living matrix. In the case of cartilage, the non-living matrix is collagen protein. So oftentimes the cells are not connected to one another directly. They're embedded in this non-living matrix. Why is blood atypical? Because the matrix is liquid rather than solid. Okay? So the fluid part of the blood is the matrix in which the living cells are located. What are our three principal types of cells in blood? Leukocytes, erythrocytes, plasma is the fluid. Platelets, which are actually cellular fragments of a intact cell. Do you know what the intact cells are that give rise to platelets? Megakaryocytes? Okay. Well, as far as defense mechanisms are concerned, which we're going to get to later on, leukocytes, white blood cells, are going to be really, really important. Okay? We're going to talk about a number of different types of white blood cells that contribute to your immunity. Platelets are important too because they produce clotting proteins, and clotting is another part of your body's defense mechanisms. Well, there are going to be microorganisms that have adaptations that allow them to react with blood cells and to overcome that part of your body's defense system. So we're going to come back to that later on. So let's say that for one reason or another, your defense barriers have been breached in some way, and you now have microorganisms growing and proliferating in your tissues. You have an infection. If enough cells are killed, then you're going to start seeing disease symptoms associated with having that particular infection. What's causing those particular disease symptoms? Well, typically what's causing them are substances that the pathogen is producing that allows them to become more invasive. We call those substances virulence factors, or you can think of them as invasion factors. Basically, these are kind of offensive weapons that allow the pathogen to spread through your tissues and overcome your normal defenses that prevent spread. What are these? These are primarily biochemicals. Many of these are secreted by the pathogen and have an effect on your tissues. And depending upon the tissue that is being affected, you will have various symptoms associated with that. So often, your symptoms of disease are due to the virulence factors that the pathogen is producing. Now, there are a couple of exceptions to that statement. Sometimes the symptoms of your disease are not the result of things the pathogens are producing, but actually are the result of things your body is producing in response to the infection. They're a result of activation of your body's defenses. When we start digging into your body's defense systems, one of the things that we're going to see is your white blood cells produce a whole array of compounds called lymphokines. And lymphokines are chemical signaling molecules. That allow your various parts of your defense system to communicate with one another and to respond effectively to the infection. An example of a type of lymphokine that we're going to talk about later on is a molecule called interferon. And it's really important that your body produces this stuff. This is a big part of your defense against infection and disease. But these chemical signaling molecules can also cause symptoms. You know how you just feel off sometimes and you know, damn, I'm going to be sick. You just have that yucky, icky feeling that precedes the really overt symptoms like coughing, sneezing, whatever. Okay? Many of those early on symptoms, those symptoms that make you feel off or give you the knowledge that you're coming down with something, are due to these defense mechanisms like the production of interferons. It's kind of a double-edged sword. So what we want to do now is to take a look at the offensive side of the equation and dig into a little bit what are some of the adaptations the pathogens have that allow them to invade your tissues and make you sick? What are some of their virulence factors? And if appropriate, how do these virulence factors contribute to a particular disease associated with a particular pathogen and the symptoms associated with that particular disease? As I said last time before break, a good diagnostician, if they take a good history, can many times work backwards from the symptoms and deduce what the pathogen is. And if you can deduce what the pathogen is, then you can oftentimes, if there's a treatment available, make a good choice about what treatment that patient should receive. It's not always straightforward. Sometimes the relationship between the symptomology and the particular pathogen is pretty complicated. But that is kind of the art of medical diagnosis. Now, that diagnosis can be aided by other information. A history can involve more than just taking, blood pressure and things like that. Sometimes you have to draw blood and use blood tests to augment the information that's in your history to really come up with a definitive and accurate diagnosis. Okay. Virulence factors can typically be subdivided into three major categories. Enzymes, toxins, and anti-phagocytic factors. And there are illustrative examples on this particular slide of those three particular categories. This shows you some enzymes up at the top. We'll talk about those first. Then, time permitting, we'll get down into the toxins and distinguish between exotoxins and endotoxins, for example. And then we'll say a few words about the anti-phagocytic factors. Bottom line, these are the substances that a good pathogen can produce that allow them to be invasive and to breach those defenses and start establishing in the tissues and cause an infection. And again, many times, because of these virulence factors, particular tissues are dying, and the death of those particular tissues gives you your particular symptomology associated with that particular pathogen. Okay. The type of enzymes that we're going to talk about are exoenzymes. These are substances that are secreted by the pathogen and they affect some type of target tissue. The target tissue that is oftentimes involved is epithelial tissue. So these are enzymes that are kind of dedicated toward breaching that outermost barrier, kind of the first line of defense that your body has against infection. Just going to give you a few examples. One type of exoenzyme that is a very potent virulence factor is the enzyme hyaluronidase. In this particular illustration up here, this shows you epithelial tissue. And one of the things that's a hallmark of the epithelial tissue is that those cells are connected to one another, forming a sheet of cells that provides an intact barrier. One of the things that is important in joining those cells together to form that intact barrier is an intracellular cement called hyaluronic acid. So that's kind of an intracellular cement that holds the cells together. So what if you can produce the enzyme hyaluronidase? A lot of species of staph can produce this. A lot of species of strep can produce this as well. What does it do? It attacks that intercellular cement and therefore causes the tissue to pull apart and the connections between those particular cells to break open, and therefore bacteria, illustrated by the green right here, can then gain entrance into the underlying tissue, which would normally be protected by that intact barrier. Hyaluronidase is sometimes referred to as spreading factor, and it facilitates the spread of the organism. Another exoenzyme that's an important virulence factor is keratinase. And this particular enzyme degrades keratin, which, remember, is that tough and soluble protein that helps to make skin a better barrier against infection. So the ability to degrade keratin is another offensive weapon that the pathogen can possess. So is collagenase. Collagenase degrades collagen and can cause that connective tissue to start falling apart. Okay, start degrading that matrix in which the cells are embedded in connective tissue. Another exoenzyme is the enzyme lecithinase. Lecithin is also known as phosphatidylcholine. Have you ever heard of that compound before? Phosphatidylcholine. It's a type of phospholipid. And where do we find phospholipids? Cell membranes, right? So this particular enzyme attacks cell membranes and causes cell lysis. When the cells break open, lysosomes also rupture. And what do lysosomes contain? Digestive hydrolytic enzymes, which then can attack other cells and cause their destruction. So this lecithinase not only kills cells, but facilitates those dying cells killing other cells. So it causes kind of a chain reaction of cellular destruction. These are only a few of the types of exoenzymes that pathogens can produce. And some of the nastier pathogens produce one or more of these substances. So you're not confined to producing a single type of exoenzyme. One of the bacteria that can cause a disease that is pretty painful is the bacterium Helicobacter pylori, which can cause ulcers, which is an erosion of the epithelial tissue that is lining the inside of your stomach. And if your epithelial tissue lining your stomach gets eroded away, that can cause pain, ulceration. If it gets really bad, if that epithelial tissue becomes completely breached, then acid from your stomach can begin to leak out into your abdominal cavity, and you can have a perforated ulcer, which is life-threatening. These particular bacteria degrade this tissue by producing a number of exoenzymes, a couple of which you can see right here. Collagenase and mucinase, that's one I didn't list right here. Mucinase breaks down mucus, and the mucus lining of your stomach is an important part of the protection against that particular acid leaking out of your stomach into your abdominal cavity. And so this Helicobacter pylori produces a number of virulence factors, including exoenzymes, that contribute to its ability to cause ulceration and stomach disease. So what we got going on here? Kind of nasty. Gangrene. Yeah. Gangrene is usually subdivided into two categories, typically referred to as wet gangrene and dry gangrene. They have a common denominator, and that is blood flow to tissues becoming blocked. That's called ischemia. And as a result, tissues start to die. And if you get massive tissue death, that typically results in this blackened necrosis that is oftentimes the hallmark of gangrene. In dry gangrene, one of the things that can cause blockage of blood flow to tissues is injury or trauma. A crushing type of injury, for example, that breaks blood vessels and diverts the blood from, in this case, the extremities, toes, the fingers, etc. What's another thing that could contribute to this dry gangrene that will block blood flow besides a crushing injury? What might be happening here? How about cold? Blood vessels constrict in response to cold. Your body's trying to divert the blood to your body core to maintain the core. And so fingers and toes can become frostbitten. And if you don't restore blood flow to that tissue, it becomes ischemic and it can start to rot basically. As these tissues start to die, this tends to become progressive because the dead tissues have their lysosomes first, and then the lysosomes release enzymes, digestive enzymes, which affect neighboring cells. So one of the things with gangrene that happens a lot is that it spreads. Okay? Wet gangrene is also known as clostridial myonecrosis. Well, we know what the necrosis part of this is right now, basically rotting tissue. What does myo refer to? Muscle. Okay? So muscle tissue is oftentimes involved in the dry gangrene. And particularly muscle protein becomes fermented. So this is a result of a bacterial infection, not typically a traumatic injury or cold, but an actual bacterial infection. Now, sometimes tissue trauma is the cause of the infection. But for this to happen, spores have to enter the tissue in order for wet gangrene to occur. The microorganism that's the causative agent of Clostridial myonecrosis is Clostridium perfringens. And this is an anaerobe. And the organism is also an endospore-producing bacterium. So let's say you have a serious injury. And that injured tissue also gets exposed to dirt, for example. Well, dirt is very rich in spores of many bacteria, including Clostridium bacteria. So these spores get introduced into this damaged tissue. The original trauma will probably cause blockage of blood flow to the tissue. And so cells that weren't killed by actual physical trauma will die eventually because of lack of oxygenation. And that original site of trauma tends to become an anaerobic environment. Blood's not getting to it, therefore oxygen's not getting to it. Any oxygen that was present is being used up by aerobic bacteria that were part of the contaminating dirt. And so we develop a pocket of anaerobic conditions. Well, those spores now can then germinate in that anaerobic environment. And Clostridium bacteria are impressive. They produce at least 11 different toxins and enzymes. We haven't talked about toxins yet. Which are all virulence factors, which basically contribute to tissue death. So they start producing stuff that really starts killing surrounding cells, including lecithinase. So this tends to become a spreading condition. If they start fermenting things like muscle proteins, what does fermentation typically produce? Fermentation typically produces gas. And what does gas lead to? Swelling. And as that tissue becomes more and more swollen and distended, what is that going to do to blood flow? It's going to pinch off blood vessels and further block blood flow. Which leads to what? More tissue death, more necrosis. The tissue death And this necrosis has a very foul smell. That's one of the ways that surgeons used to diagnose gangrene, is that they would go up and take a whiff of the patient's tissue, and if it had a particular smell, which was really a nasty smell, they could really smell gangrene. So as these bacteria start spreading throughout the infected tissue and start killing healthy cells that weren't part of the original injury, because of the lecithinates and other virulence factors and toxins they produce, this becomes a progressive infection. What are your options for treating it? Amputation of healthy tissue. Since this is a bacterium, what's another possible treatment you could use? Antibiotics, okay. Since this is an anaerobic bacteria, what's another possible strategy? How many of you have worked with Dr. Dana before?
Oh, the gas chamber they have, the vacuum.
Hyperbaric oxygen treatment. What does baric refer to? Pressure. Okay, so this is oxygen under pressure. What does that cause? That causes the oxygen to infiltrate into the tissues more effectively. And the oxygen will what? Generate reactive oxygen species. These anaerobes don't have the detoxifying mechanisms to deal with those ROSs. And so that hyperbaric oxygen can be useful in augmenting some of these other treatments. Gangrenous areas. If you don't treat it, mortality rate exceeds 40%. And typically the results of death are kidney failure, shock, tachycardia, which is increased heart rate, but it's not something you want to mess around with. So in the case of the wet gangrene or clostridial myonecrosis, the virulence factors that those particular pathogens produce contribute directly to the spread of the disease and the symptomology of the disease. Well, next time we'll talk about some different exoenzymes. Some exoenzymes react specifically with components of atypical connective tissue and the blood. So we'll talk about some hemolysins, coagulase, and some of the other exoenzymes that interact with blood.